Welcome to section 4.4, Fermentation. In this section, we'll examine how cells deal with energy production when oxygen is unavailable or in short supply. We'll focus on fermentation, a process that keeps glycolysis going when oxygen is limited or absent. It may not be efficient, but it's a powerful adaptation for survival. By the end of this section, you should be able to describe the fundamental difference between anaerobic cellular respiration and fermentation, and explain lactic acid fermentation in animal cells and the conditions that trigger it. Keep these goals in mind as we explore how cells adapt to low oxygen environments. Imagine running up a hill and suddenly your muscles burn. You're trying to catch your breath. That's fermentation at work. When oxygen runs low, cells still need to make ATP. Glycolysis can do that, but it needs NAD+. Fermentation helps regenerate NAD+ from NADH, so glycolysis can continue. It's not efficient, just 2 ATP per glucose, but it keeps energy flowing during an oxygen crisis. Anaerobic respiration still uses an electron transport chain, but the final electron acceptor isn't oxygen. Instead, certain prokaryotes may use nitrate, sulfate, or even carbon dioxide. This process is more efficient than fermentation because it still creates a proton gradient to power ATP synthesis, though it generally produces less ATP than aerobic respiration. Fermentation, by contrast, skips the electron transport chain entirely. It uses an organic molecule, often pyruvate or a related compound, to accept electrons from NADH. This regenerates NAD+, so glycolysis can keep running. Fermentation is simpler and produces only a small amount of ATP, but it works in environments where oxygen or other electron acceptors are unavailable. In animal cells, like your muscle cells, lactic acid fermentation kicks in during intense activity. When oxygen can't keep up, pyruvate accepts electrons from NADH, forming lactic acid and regenerating NAD+. This helps your muscles keep working, but it also leads to soreness and fatigue. Once oxygen returns, lactic acid is either broken down or sent to the liver to be recycled via the Cori cycle. It's a short-term strategy for survival. Alcohol fermentation is used by yeast and some bacteria. First, glucose is broken down into pyruvate. Then, pyruvate becomes acetaldehyde and carbon dioxide. Finally, acetaldehyde accepts electrons from NADH, forming ethanol and regenerating NAD+. This process helps the organism survive and is useful to us in brewing, winemaking, and baking. Unlike fermentation, anaerobic respiration involves an electron transport chain and occurs mostly in prokaryotes. These organisms use alternative molecules like nitrate or sulfate instead of oxygen as final electron acceptors. This process produces more ATP than fermentation, but less than aerobic respiration. You can find these bacteria in oxygen-poor environments like swamps or deep soil. Fermentation allows cells to regenerate NAD+, so glycolysis can continue without oxygen. There are two main types: lactic acid fermentation in animal cells and alcohol fermentation in yeast and bacteria. Both regenerate NAD+ to sustain ATP production from glycolysis. We also discussed anaerobic respiration, a more efficient process used by some prokaryotes. These strategies help cells survive and function in low oxygen or oxygen-free environments.